and prospective meta-analysis methods group at the University of Sydney. Research focuses on collaborative methodologies to enhance evidence synthesis in clinical research. And together with Lene Seidler, formerly a biostatistician at the Clinical Trial Center at the University of Sydney, who has just moved to Universitätsmedizin Rostock in Germany, they are developing an innovative tool to detect untrustworthy medical studies, ensuring a reliable evidence base for clinical guidelines and practice. And we are very interested in hearing about this more.
Thank you very much for that introduction. I'm just confirming that you can see my slides okay.
Yes.
Thank you. So yes, hello and guten Tag or gute Nacht for me here in Australia. I am really honoured and excited to be here and would like to thank the Einstein Foundation for the opportunity to present this work today on behalf of my team at the University of Sydney. So there is an integrity crisis in research. And while I mainly talk about my field, which is medicine, we also intend to apply our work across disciplines. So last year alone, more than 10,000 publications were retracted due to integrity issues. And we've seen a proliferation of articles on cases of fraud, research misconduct, and research integrity issues, some of which are shown here. Estimates about the extent of the issue vary, but experts say that about more than 20% of published research may be affected by research misconduct. And the consequences of this can be really severe and in direct contrast with the underlying principle of medicine, that is, first, do no harm. So if we think about, we can consider untrustworthy research to be a pollutant, which flows into our river of evidence, contaminating our evidence base, and then this flows on to inform guidelines, policy, and treatment decisions. And a recent example demonstrating these potential consequences is the case of ivermectin for the treatment of COVID. So during the pandemic, millions of COVID patients globally were administered the drug ivermectin based on a review of studies showing it could increase survival by 51%. However, it was later discovered that several studies underpinning this evidence were fraudulent or had major integrity issues. And when these studies were excluded from the analysis, there was no longer any evidence of effect. So here we have patients that are administered a drug without any benefit, but with known side effects. And this led to a shortage of ivermectin for other approved uses. This also led to public mistrust in science and wasted resources, which could have been directed elsewhere. In the words of James Heathers, this crisis represents an existential threat to the scientific enterprise. So we have a crisis. What do we do about it? So there are emerging tools to assess integrity of studies once they have been published based on summary data and information such as ethics approvals and trial registration. However, many integrity issues can only be detected by scrutinizing raw individual participant data. So by that, we mean row by row data for each participant within a study. And as you can imagine, this enables more detailed checks. And the power of having these data available was demonstrated by editor John Carlisle. So he examined trials submitted to his anesthesia journal and found that when raw data were not available, almost all studies appeared to be trustworthy, as indicated in blue. However, when raw data became available, he detected a massive 44% of studies that included flawed data. So we know that IPD are important, but there were no standardized tools to assess the integrity of trials using these raw data. So we set out to address this gap. We aim to develop, validate, optimize, and disseminate an individual participant data integrity tool. So what have we done to date? Well, as part of my PhD, I led development of an initial tool. This is based on a review of the literature and some preliminary mapping, a discussion among advisory group about important items, some pilot testing and preliminary validation. And the tool was recently published in Research Synthesis Methods. since publishing, I've received an influx of requests from other researchers, journal editors, and publishers who are interested in applying the tool. And this really made me realize that there's a potential for a much broader application and benefit than I originally foresaw. So, and also the need to make the tool more accessible and fit for purpose for a variety of different stakeholders. So briefly, the pilot tool has 3 components, an instructions and decision guide, a rating sheet, and an automation script to semi-automate some of the checks. And it includes domains at the publication level because these are still important, but it also includes, as the main point of difference, the additional checks we were able to do with individual participant data. So this is what we've done to date. What will we do next? So our key objectives now, we will further validate the tool to ensure it can accurately identify untrustworthy studies. And to do this, we'll use a range of data sources, including fake data sets provided to us by journal editors and data sets generated using artificial intelligence. We'll also further refine and automate items within the tool. So currently we have created an R script for this purpose, but this requires some coding expertise and therefore limits accessibility. So we are working with computer scientists to optimize this. So essentially, we'll be looking more closely at these IPD domains shown here in green, and we'll work to determine the minimum number of domains that we can include to make the tool feasible, but while still being able to predict integrity issues precisely. And we'll do this by looking at test theory characteristics, so sensitivity and specificity, but also things such as ease of use and feasibility. Initial feedback from journal editors was also that a lack of time and expertise would limit their implementation to implement the tool in its current format. So we plan to develop a streamlined, easy-to-use interface in line with FAIR principles and in consultation with these key stakeholders to ensure it's fit for purpose. And this user-friendly interface might look something like this, where the user would enter key information, such as variables they might expect to be correlated, and then it generates a report tailored to these different users. We also aim to disseminate the tool widely, and we have a well-established international network of key stakeholders who have pledged their support. For instance, Cochrane and various publishers and journal editors. And these have either been involved in the initial development of the pilot tool or have contacted me because they're interested in applying it. So we also plan to move beyond medicine. We're closely connected to researchers across a range of disciplines who have shown interest in applying the tool. So we wish to work with them to transfer the tool to them. And lastly, we want to continue to update the tool to keep apace with emerging methods. So the tool is not intended to be static. It'll continually be updated to address emerging challenges, for instance, those that might be posed by developments in AI. Now, my description of the tool to date may have seemed a little bit abstract, so I'd like to share some concrete examples of what this sort of tests might look like or checks. And the first one is regarding data patterns. So we can see in this table here, we have the birth weight of 20 different infants. At first look, it might seem okay, but upon closer scrutiny, we can see that the first 5 rows of data have simply been copied and pasted across infants, something that does happen and is an indicator of fraud. Another thing we look at is correlations. So are expected correlations present? So in trial A on the left, we've got gestational age at birth on the x-axis and birth weight on the y-axis. And as expected, as the baby is born closer to term, we have a higher birth weight. But in a real-world example that we came across, there was absolutely no correlation present whatsoever. The publication looked fine, but the IPD or raw data enabled us to see this. This raised red flags about the integrity of the data, and we excluded this study from our meta-analysis. So who could use the tool? We envisage several different stakeholders that have expressed an interest in using the tool. Publishers and journal editors may wish to use the tool to guide decisions on whether a manuscript should be considered for publication or whether perhaps a retraction or expression of concern is warranted for a publication. Systematic reviewers could use the tool to determine whether a study should be included in their meta-analysis. And research institutions could investigate allegations of misconduct among staff. So this proposed project will take our pilot work and leverage our collaborations to develop an innovative, validated, easy-to-use tool for widespread application that will filter out untrustworthy studies from the evidence base, leading to more reliable evidence to inform guidelines, policy, and practice. And this will reduce research waste, protect patients, and foster trust in research. So we can consider the big picture of the evidence ecosystem. We can really use this crisis as a catalyst for change. So our integrity tool can help to raise awareness about integrity issues, pinpoint where these problems arise, and drive systemic change to address these problems. So while our integrity tool is one part of the solution, we really need to combine it with systemic changes to prevent this pollution happening in the first place. So we need a cultural shift to deter misconduct and incentivize best practice. So lastly, I'll leave you with this quote: "The world is a dangerous place to live, not because of the people who are evil, but because of the people who don't do anything about it." So let's do something about this and safeguard the integrity of research. I'd like to acknowledge my team, and I'd like to thank you. Dankeschön. Happy to take questions.
Kylie, thank you very much. I guess Lene might also join the discussion now. And